Greetings everyone. So we are continuing from where we left off. So today we're gonna talk about blood grouping, but before we go there we need to understand just a little thing. We have to think about the types of dominance before we can chat about blood grouping. So we have the complete type of dominance, we have the incomplete type of dominance, and we have the core dominance. So what do you need to know about the complete dominance first? So what you need to know here is that there is a dominant and a recessive allele. A recessive allele. Sorry, let me rub this off. Recessive allele. So with complete dominance, it is whereby there is both a dominant as well as a recessive allele. As a result, the dominant allele will express itself in the phenotype if it was there on the genotype, and the recessive one will only be expressed if it's a homozygous condition. In the past lesson, we talked about homo as well as heterozygous. An example here of complete dominance would be breeding a red and a white flower, and all your flowers become red, or most of them become red, and those that are white are in homozygous condition. So that's all about complete dominance. What you have to know is that there is a dominant and a recessive allele. That's the key for complete dominance. Then when you go to incomplete dominance, here no allele is dominant over the other. No allele is dominant over the other. Here there is no allele that is dominant over the other. As a result, when those alleles are combined, an intermediate phenotype arises. For example, with red and white, if red is not dominant over white and white is not dominant over red, then when they are combined, an intermediate phenotype arises. Let me just write intermediate phenotype. So if it's red and white, then maybe a pink flower. If you're crossing a red flower and a white flower, then you get a pink flower. Reason being, red is not dominant over white, so they can't be all red or most of them be red, and white is also not dominant over red, so they can't be all white or most of them be white. That's what we term as incomplete dominance, because no one dominates the other. Then we represent this in this way. We say, maybe if it's red and white, it's R, W, they will all be capital letters. For core dominance, they are equally dominant. Equally dominant. As a result, they will be both expressed in the phenotype. For example, same example, it's a red and a white flower, then a resultant flower will be a flower with both red and white patches. So that means they will both there will be both red and white in that flower. They will be both expressed because they are equally dominant. That's all about types of dominance. I will move to blood grouping, which is the main thing we're going to talk about today. So with multiple alleles, actually with blood grouping, there is a concept of multiple alleles. So we have to understand what are multiple alleles. It is when we have more than two, not more than one, more than two alleles of a gene for a particular feature. So if you have more than two alleles of a gene for a particular feature, then that's termed as multiple alleles. What do I mean by that? For example, for eye color, just a lousy example, or for skin color, maybe you will have maybe two alleles for that. Maybe the one that will make you black and the one that can make you white, for example. Then maybe if the black is dominant over white for any reason, then you'll end up being black. That's just a lousy example, but I'm trying to show you that before we did an either or situation whereby we said it's either this or that, but in this case it is when we have like multiple things to choose from to result in different phenotypic expressions. Because we can go back to the color of the flowers we used in the first example. We said red or white. That means if it's not red, it's white at a phenotypic level. Then at a level of alleles, it's an allele for red, it's an allele for white. So like it's an either or in terms of the alleles. But now we will have multiple alleles. That means we will have more than just two alleles for that gene, and they are called multiple alleles because they are found on the same locus in a chromosome. So that's why they are not considered different, different alleles. I mean, yes, different alleles at different genes, but because they are found on the same locus in a particular chromosome, then they are called multiple alleles. And the most important part is that they control one feature as much as they are multiple. That means as much as it's monohybrid in terms of crossing, because it's just excuse me, one feature, but we are choosing from a variety of options. Apologies for that, my voice isn't so fine. So in humans, we find multiple alleles when we are doing blood grouping, which is what we're doing right now. And the importance of that is that it increases variation, because if we have a lot to choose from, that means also we will have a variety of phenotypes or phenotypic expressions of those alleles. But this might not make sense at this point in time, but I'm hoping as we move along it will do make sense. So with alleles for blood grouping, we've got IA, we've got IB, and we've got small letter I. So these are alleles for blood groups. So you see, that's why I said multiple alleles, because it's not like either A or B, but it's A, B, or I. So it's like multiple because there is more than two alleles coding for a specific phenotypic expression. So your question might be, then what are the possible phenotypic expressions? Then we have 4 possible blood groups. which are one, blood group A, blood group B, blood group AB, and blood group O. So we've got those possible blood groups. Then what are the possible sort of alleles for them? Then for blood group A, you can either have IA, or you can have IAI. I'll tell you why. For blood group B, you can either have IB, which is homozygous, or IB, then I. For blood group AB, you can only have IA, IB. For blood group O, you can only have the two small letter I. Why is this the case? Remember, in the previous part of this lesson, when I talked about dominance, so what you need to know is that A and B equally dominant. So equally dominant, you know, most co-dominant. Co-dominant. Let me cancel out that equally dominant, co-dominant. So when we talked about co-dominance, we say they are equally dominant. So as a result, they will both express themselves in the phenotype. So you see now it's a combination of A and B. But we are not saying it's blood group A, and we're not saying it's blood group B, but we are saying it's blood group AB, because they are equally dominant. That means they both express themselves in the phenotype. Then if we think about another thing, we have to know that both A and B are dominant over small letter I. Why do we say that? You see here, there is B as well as I, but we say the phenotype is blood group B. So that means B is able to mask off small letter I. So B is dominant over that small letter I. Even for A, we say it's blood group A, but in terms of alleles, we've got both A and small letter I, but we say it's blood group A. That means A is dominant over small letter I. So those are the things that you have to know regarding blood grouping. If we had time, we would move to like crossing, but due to time, I'm just gonna tell you a few more things you need to understand before I mess up my slides. Okay, so what you need to know is that each person can have a maximum of 2 alleles. As much as we've got 3 alleles, but each person can only have 2 alleles. Okay, this doesn't want to write. It seems like my pen doesn't want to work, but it's fine. I think I've written what I wanted to write. So yeah, that's what we have to know, that as much as we have a combination, we have like 3 alleles, but each person can have a combination of only 2. As you see here, it allows me to point. As you might see here, like they go in 2s. So you can only have a combination of 2 as much as there are 3 options. That's why they say it increases variation, because you can be A, someone can be B, someone someone else can be AB and someone else can be O. So we are not only confined within just two options, just like in the past when we didn't do multiple alleles. I guess I have to stop right here, and we will chat again when we I don't know what we will be doing, but I might just stop right here.